Hello and welcome back, future scientists. I'm Mr. K with Mississippi Public Broadcasting. We're going to continue looking over some state test review questions. So teacher, if you could, go ahead and pass this worksheet out to your students. You all take 10 or 15 minutes to work on it. When you're ready for the answers, come back to me, unpause the video, and we'll do the questions on the board behind me. All right, I'm sure that didn't give y'all any problem, but just in case it did, I'm going to do the questions on the board behind me, so thanks for following along. Let's go ahead and get started. Okay, this says, the diagram shows mutations that have been observed in a mouse genome. We have the wild type genomic DNA sequence. This just means it's the normal one, so this is what it should be like. And then we have the mutated genomic DNA sequence. So there's been a mutation here, okay? It's asking us to, this says, using the mRNA codon chart, what is the most likely effect of this mutation on the resulting protein produced? So you can see here, this is where our mutation occurred. It's a single nitrogen base. That G was replaced with a T, okay? The answer here should be D, okay? What's going on is we're going to produce a different amino acid. This right here, the GGC, this one right here, when we turn that into mRNA, it's going to be CCG, which is going to be this one right here. The way you read this is you always start on the left. You can see this is our first position. It's a C, and then the second position is a C, and then the last one is a G. Okay, and this is telling us that it's going to make an amino acid called proline. Okay, what happens when this mutation occurs, we're making a different mRNA sequence. Instead of this, we're going to get CAG, okay? And what that means is we're going to make this amino acid instead. And so we're making a different amino acid. This amino acid is going to replace that amino acid, and that's probably going to change the protein produced, okay? So that's kind of how we could have tackled that question. Okay, let's move on to the next one. This says, a student uses a Venn diagram to compare the mechanisms of transcription and translation during protein synthesis. The incomplete diagram is shown. Write the number 1 or 2 next to each descriptor to identify whether it should be grouped with transcription, we're going to use the number 1 for that, or translation, and we'll use the number 2 in the Venn diagram. All right, the first one says, has mRNA as a product. Okay, well that's definitely transcription. Remember that during transcription, we use our DNA to make messenger RNA, right? So that's the first part of this whole protein synthesis is DNA to messenger RNA, and that happens during transcription. The next one says occurs in ribosomes. This is going to be 2, okay? During translation, the mRNA moves out to the cytoplasm, it finds a ribosome, and it's going to start building the protein amino acid by amino acid. That happens at the ribosome, and that's what's happening during translation. The next one has polypeptide as a product. Okay, this is also going to be translation. In translation, we go from messenger RNA to proteins. Remember that polypeptide sometimes is used interchangeably with the word protein, but through this process of translation, we're making a polypeptide. So that's going to be number 2. And then the last one there says occurs in the nucleus. Well, transcription occurs in the nucleus. Remember, DNA is in the nucleus, and we're copying the DNA, and so the mRNA is made in the nucleus. Okay, so that should have been the correct answer for that one. Great work. Let's move on to the next one. This says, the model shows structures within the nucleus of a cell. Which 2 statements best explain the relationship between these structures? Okay, for this one, here are the answer choices that I like. Letter D, this one says, the genetic code specific for a protein is carried in the genes that make up the DNA molecule. The other one I think is correct here is F, okay? This says the DNA molecule is made up of nucleotides, and the gene is a section of DNA that contains nucleotides that code for a specific protein. A lot of these answers are just using vocabulary words, and you need to be able to use them the correct way. D and F are the ones that are correct here. All the other ones aren't really using the vocabulary words in the right way. Okay, great. Let's move on to the next one. This says, a student is researching the benefits and risks associated with transgenic organism in agriculture. Record an X in each row of to identify whether the statement describes a benefit or a risk associated with transgenic organisms. Remember, transgenic organisms means that we have altered the DNA or the genes of an organism in some way. For this question, it wants us to, for these different options, say whether that might be a benefit or a risk. The first one says cross-pollination with native organic plants. This should be a risk. Y'all might remember talking about invasive species and how sometimes if a species is not native to a food chain, it can cause some disturbances. This is very similar to that. The next one says decrease in the consumer price of meat. Well, that would be a benefit, right? If we're able to make more food and make people more food secure by decreasing the consumer price of meat, that means more people are going to have food. That's going to be a big benefit, right? The next one says human hormonal effects from consumption. Okay, well, that one's probably going to be a risk. We don't really know how these transgenic organisms might affect a human, and so if it does have an effect, it's probably going to be a risk. And then the last one says increasing crop tolerance from pesticides. That's going to be a benefit. If our food isn't getting destroyed by pesticides and these other insects, that's going to be a good thing for us. So we have some benefits and some risks for these transgenic organisms. Great work. Let's move on to the next one. Okay, students are comparing the processes of transcription and translation in a cell. Record the letter of each process feature in its associated column in the chart. Okay, so here we have A, B, C, D, and E, and we're going to put them in either the box for transcription or the box in translation. Let's see if we can sort these out correctly. So A says occurs at the ribosome. Okay, well remember that in translation, the ribosome plays a huge part in translation. A ribosome and an mRNA come together and starts to build that polypeptide, right? The next one says occurs in the nucleus. Okay, well, transcription occurs in the nucleus. Remember, transcription happens first. We're copying the DNA. The DNA is in the nucleus, so if we're copying it, we have to be in the nucleus as well. The next one says reads RNA to make a protein. Okay, well, this is going to be C. Okay, during translation, we take the mRNA we made in transcription and we use it to make proteins during translation. Okay. The next one says uses DNA to make RNA. That is transcription. That is exactly what's happening. We use DNA to make messenger RNA in the nucleus. That's a pretty good overview of what transcription is. And then the last one, links amino acids together. Remember that amino acids are the monomers of proteins. So if we're linking them together, we're building a protein or we're building a polypeptide. And so that is going to describe translation. So that's how we should have sorted those letters. Okay, great work. Let's move on to the next one. Okay, this says adult somatic cell nuclear transfer, also known as cloning, may potentially be a method to recover populations of endangered species. Recently, cloning was used on the last surviving cow of a rare breed from New Zealand. What is the strongest ethical and moral argument against continuing research and experiments in cloning endangered species? A really good answer choice for this one, I think, is C. Okay, this has possibilities that process could be used on organisms for personal preference. Okay, the other ones aren't quite as strong moral arguments against using the experiment. Okay, so this is going to be our best answer, and we'll go ahead and move on to the next one. This has an original segment of DNA and a segment that has been mutated due to exposure to UV radiation are shown. This says which sequence represents the mRNA produced during the transcription of the mutated DNA segment. Okay, so what we really want to notice here is that we're taking this mutated DNA segment and we're going to make messenger RNA out of it. Okay, when you're making messenger RNA, there are some base pair rules that you want to remember. Okay, those base pair rules are that A is going to pair with U, T is going to pair with A, G is going to pair with C, and C is going to pair with G. And so I think B is our best answer choices here. If you use those base pairing rules, this is what you should have come up with. Just as a reminder, when you have mRNA, you don't have the letter T. You use uracil instead of thymine. So anytime you're making messenger RNA, you can cross out any options that have a T in it. So we knew right away that that wasn't the right answer. Okay, great work. Let's move on to the next one. Okay, this says, refer to the mRNA codon chart below. Okay, which statement best describes this mutation? And it's talking about the mutation that we were just looking at earlier. So here's where the mutation occurred at. This third codon is where the mutation happened. You can see that this is a C instead of a G. Okay, and the answer for this one is that C is going to happen, and we'll look over that. The third amino acid in the protein would change from one amino acid to another, which may affect the shape and function of the protein. So that's all the questions that I have today. Thank y'all so much for following along. Your teacher has some other work for you to do, so make sure to give that your best effort, and I hope everybody has a great day.